Hello, everybody, and welcome to our third BioPharma panel this week. Pleased that you've given us some time and consideration during your busy schedule. My name is Jay Blackwell. I'm the Executive Director of the American Heart Association chapter here in Boston. Excited to be with you all today and excited for you to hear our speaker and an amazing opportunity that's here in Boston. Dr. Stanley Shaw is the Chief Science Officer for One Brave Idea, an initiative funded by the American Heart Association, by Verily, AstraZeneca, and Quest to develop new approaches to understanding coronary heart disease. He leads the research efforts in digital health, bioinformatics, the gut biome, and non-traditional streams of patient-reported data. He also serves as the Associate Dean for the Executive Education at Harvard Med School and is a practicing cardiologist at Brigham and Women's Hospital here in Boston. Previously, Dr. Shaw co-founded the Center for Assessment and Technology and Continuous Health at Mass General, and his team led the development of one of the first research kit iPhone apps for type 2 diabetes in partnership with Apple. Dr. Shaw, thanks for joining us today. And with that, I'll let you take us on a wonderful journey. Thank you much.
Great. Thank you, Jay. Thanks to the AHA for the invitation. Let me share my screen. Is that looking okay? Oh, everyone's dark, but yell if it's
We're good to go.
Good to go. Great. So thanks for joining, and I hope that we can have an interesting conversation. The title is Beyond Genomics, an Ecosystem for Cardiovascular Innovation. And I think it's a perfect time to actually have a slightly broader discussion, not just genetics, not just genomics and cardiovascular disease, but what is the broader societal and ecosystem landscape in which these kinds of research innovations ultimately have to take root. And there will be a couple of polls in this. We'll try that out to try to get some engagement among the audience. Just a couple of disclosures, as you can see somewhere above my shoulder, that Verily, AstraZeneca, and Quest are certainly supporters of the One Brave Idea initiative. We're very grateful for their partnership. And my spouse works for Novartis Institutes for Biomedical Research, albeit not in heart disease and in the field of cancer. So the title of our One Brave Idea project is Redefining Coronary Heart Disease at the Edge of Wellness. The project leader is Dr. Callum McRae. He is a professor of medicine at Harvard Medical School in the cardiovascular division, a cardiologist, and also a vice president for innovation at the Brigham. He sends his apologies. He would have loved to be here to give these remarks himself, but he had committed months ago to give medical grand rounds at another institution. So he does send his regrets. The goal of our project is to shift the lens of focus for heart disease from middle age, where risk factors become apparent, diagnosis and treatment efforts intensify, to a couple of decades upstream, and to try to enable earlier detection and interventions. When we do this, we recognize that we are benefiting from the tremendous legacy and ongoing work of the AHA to implement the findings from basic and clinical research in the real world, in the lives of patients. Here is the Life Simple 7 about different kinds of medical and lifestyle factors that are the keys to cardiovascular health. And, you know, this legacy and testament from basic science to implementation in the lives of people is really a testament to the work of the volunteers and supporters of the AHA, like all of you. And so against that backdrop of success, these are some of the questions that guide our efforts at One Brave Idea. How can we identify causal mechanisms early in the development of heart disease and enable earlier intervention and diagnosis? How can we leverage genetics and the advances in genetics more powerfully to develop therapeutics and increase our understanding of disease? And there are a couple of other, in red, kind of broader scientific, but other almost scientific and cultural aspects to our work. How can we make better use and learn from the data generated in clinical care? This idea of a learning health system, I think, is really gaining traction. It's very hard to implement. And the related hope is that if we can do this, we can start to break down silos that separate patient care and research. And these silos also keep, erect barriers that prevent many people from participating in the research enterprise. And in so doing, we hope we can strengthen connections between patients, patient care, care delivery, and therapeutic development. So here we are in Boston, and it's a time of unprecedented innovation, thanks in part to novel platforms and novel therapies. Here are the coronavirus, that are starting to show us the light at the end of the tunnel. And it's just a reminder to me to acknowledge, the work that many or all of you are doing to get everyone through this pandemic, whether it's through your official work, your community, your family. But there's a longer history of tremendous innovation in recent years and really tackling some long-standing genetic diseases that have eluded therapies. This is a photo of a young man with cystic fibrosis, and he's holding the copy of Science magazine where he was photographed as a young boy, right? The gene for cystic fibrosis was one of the first disease genes to be cloned in 1989. And 23 years later, Vertex came up with their first small molecule therapy against cystic fibrosis. They've gone on to release other small molecules. And the interesting thing for this discussion is that these small molecules not only treat cystic fibrosis, treat based on their genetic profile of mutations in the CFTR gene. Another one also in the Boston, Cambridge area are RNAi therapies. On the left, you can see the actual basic science papers that led to a Nobel Prize in this area. Those papers are from 1999. And now in 2018, we had the first ever RNAi, inhibitory RNAi therapies from Alnylam that were approved, and they've also gone on to approve more. This is CAR T therapy, right? Chimeric antigen receptor T cell therapy for cancers. This is Emily Whitehead. She was the first girl in these clinical trials. She is now 8 years cancer-free, having stopped all therapies. And that's out of Novartis. This is the last one. I could go on, but this is the last one I'll do. Sickle cell therapy, I wanted to mention because it was the sickle cell gene, the hemoglobin gene, was really the very first disease where a molecular cause and a single gene was identified. The paper I am showing you is from 1949. And on the right panel, you can see a wonderful quote from a participant in a trial at the NIH. And it just speaks to the power of gene therapy, in her case, to cure this disease. And on the bottom right is the paper from Vertex and CRISPR Therapeutics using a slightly different approach, genome editing, also to treat sickle cell. So these are not FDA approved yet, but the progress is really, I think, qualitatively different than what's come before. So even though we have wonderful progress, you all know that this is still a healthcare system that is under pressure. And the last year really has been unlike anything I think most of us have ever seen. And we've had a syndemic, right? Not just a pandemic, multiple interacting crises of the coronavirus. And I think the coronavirus helped lay bare vulnerabilities in our healthcare system, from inequities in healthcare for different patient populations to financial models of care delivery. And this is not at all an abstract concept. This is a paper actually from my home institution at the Brigham and Women's Hospital. I wasn't involved in this. This was really a house staff led or heavily involved effort. And you can recognize, some friends and prominent AHA investigators in this author list. They basically looked at and tried to quantitate if people came into the emergency department with heart failure, were they admitted to the cardiology service or were they admitted to the general medical service? Both of these paths are open. You might imagine that the cardiology service, we would like to think maybe in our vanity that being admitted to cardiology might have slightly better outcomes, but really the general medicine service at the Brigham should be really quite good. And when they looked, 2,000 patients, Black and Latino patients, were admitted to the cardiology service at a statistically lower rate. And there were some downstream outcomes from this. If you were admitted to the cardiology service, you were less likely to get readmitted for heart failure within 30 days, right? This at present defies easy explanation or easy remedies, but I think it's a powerful reminder that we have to think intentionally about these efforts and everything we do. And, at One Brave Idea, we do, in addition to the science, we are starting to think with intentionality at a very early stage of the work, how the multiple audiences and stakeholders might receive or perceive value in what we're doing. I think it's important for researchers and people involved at every step of this cycle of innovation to think about the multiple audiences or stakeholders who will be affected by any kind of scientific or care delivery innovation, and to navigate these sometimes competing perceptions of value among the different stakeholders, because that will really ultimately determine whether it's widely adopted, whether your innovation is sustainable. And so that's my segue into genetic testing in cardiovascular disease. Most of the success stories and new therapies I began with were monogenic diseases, right? One gene, a mutation, is enough to set you down the path of having the disease. We know that cardiovascular disease is much more common genetically, and it's heavily influenced by the environment. And so the current estimate is that, cardiovascular risk is maybe 20% genetics, 80% everything else. So that's environment, systems, lifestyles, and behavior. With that in mind, let's talk a little bit about genetic testing in cardiovascular disease. There certainly are tests for specific genes that can be mutated and contribute to all of these conditions I've listed: diseases of the heart muscles or cardiomyopathies, diseases of rhythm, arrhythmias, diseases of the aorta, other vascular diseases, and of course, our most famous story, familial hypercholesterolemia related to LDL cholesterol. And this is from an American Heart Association scientific statement last year. And the details don't matter too much, but these are specific disease entities in the left-hand column. They correspond to the broad categories I had in the previous slide. And on the right are the alphabet soup of all the different genes that, there's at least some evidence that testing can be helpful. And certainly, the testing, if you find something, it can be useful for instance, diagnosing people with very rare syndromes that really defy conventional classifications, this really can be life-changing for a family and for an affected child often. And then the more typical kind of affecting diagnosis, family screening, risk stratification, and clinical management. However, here I've highlighted the only genes for which there's a specific therapy that's tied to that molecular defect. So that's transthyretin for certain types of amyloidosis, the LDL receptor for cholesterol or LDL lipid, and then PCSK9, which also is a powerful determinant of LDL levels, right? Everything else, the treatment is, well, maybe it's timing of surgery, or maybe it's a general risk stratification for arrhythmia. Maybe you might put in an implanted defibrillator. So it's not, gene leads to a specific treatment, except for these rare instances. The other thing I'll say is that the clinical assessments that would lead someone to say, gee, maybe we should do this genetic test, these are relatively generic clinical assessments. A lot of them are anatomic imaging, right? Ultrasounds or the echocardiogram, CAT scans, MRIs. We've made tremendous progress with contrast agents and the like, but it's anatomic, maybe a tiny bit of functional imaging. The tried and true EKG that was invented, over 100 years ago, and some blood biomarkers. Again, I'm going to return to this, but there is a little bit of a disconnect in that our ability to test genetic mutations and understand the genetic contributions to cardiovascular disease is much greater than our ability to measure it, right? To diagnose it in the tests and assessments that we usually do. Just a brief word about polygenic risk scores. Some of you may have read about this in the news or be thinking about using it. This is the other extreme from one gene, one disease. This is the idea to say, we know that, let's say, heart attack risk involves many, many genes. So we're going to take, in this case, 6 million markers, sequence markers from across our 3 billion bases in our DNA. So 6 million of those markers, and we're going to calculate a score. The higher the score, we know we can calculate these scores so that the higher your score, the higher your risk of having a heart attack or developing coronary artery disease. The interesting thing is, so if you look at people at the extreme, so if you look at that bell curve, those dark blue shadings, those have found that, for instance, if you're in the top 0.5% of this distribution, so you're very unlucky, your polygenic risk score is worse than 99.5% of the population, your risk of having a heart attack is 5-fold the population average. So what this means is that by summing up this kind of very distributed risk score across millions of spots across your chromosome, you can identify a small subpopulation whose risk is comparable in magnitude to one of those single gene disorders that we've talked about before. And so it is going to bring more people into the fold of knowing that they are high risk. The other thing about these people is, if you take that slice of that unlucky 0.5% or that unlucky 2% with the highest, worst scores, their LDL is actually unremarkable, right? It's not high LDL. And in fact, this polygenic risk score does not correlate with traditional risk factors. So that's promising scientifically. Unfortunately, we're at the very beginning. We really don't know the mechanisms why these people with these high polygenic risk scores are at higher risk. The intriguing things in terms of how this will be used, even though we really don't fully understand how to use these in clinical practice, is that this is available at birth, right? I mean, these are your germline DNA, So you could get this polygenic risk score on a baby, and it's their polygenic risk score for life. And the other market development is that companies like 23andMe have started to offer variations of this, simpler, maybe not 6 million, maybe 1,500 markers. And they offer one for type 2 diabetes now. I don't think there's one for coronary disease, but it's exactly the same concept. So this is lurking on the horizon, and it remains to be seen how best to use it. But again, to return to the vulnerability that I mentioned in the earlier slide, let's say you take one of these tests and you're found to have a high polygenic risk score. What then? And then again, the treatment is powerful, but relatively generic in terms of mechanism. You'd be urged to do lifestyle modification, and maybe you'd be urged to start a statin. So again, there's this mismatch. And so to sum up, I think there's, certainly great progress, and much of it supported by the American Heart Association on our molecular understanding of disease, cheaper sequencing platforms, and leading to an accelerating use of different kinds of genetic tests. These tests are still not widely used in clinical care, and there is this mismatch that I think is a lost opportunity. Okay, so now perhaps we can try this poll question. Meg or Emily, there it is. Wonderful. So the poll question is, what is one measurement or test that you or your doctor follow related to heart disease risk? And so go ahead and select multiple choice. So I think you can choose more than one. It's totally anonymous. Let's fire that off. We'll let that run for a little bit. Great. All right, so I think we can close the poll. And this is what you would expect, right? All of these are in the AHA's Life's Magic 7, blood pressure, weight, lipids, etc. None of you said other, which I think is interesting. The famous Paul Dudley White, right, of early cardiology and AHA lore, mentioned all of these factors, right, in his lifetime, in his practice, in his life, just to make the point that these are very old measures. And so there's an opportunity there. Okay, so nobody said other, but perhaps, some of you are doing something digital, maybe an app or a wristband for exercise. Wonderful, thank you very much. Let's close that poll. Okay. So, the way we think of assessment, measurements, and risk currently, we come up with a predicted score, and there's empiric knowledge that, the people with a certain predicted score, their actual score, looking at tens of thousands of people, actually correlates pretty well. So then we go on and we take some measurements and we say, okay, we think your point risk estimate at this stage of your life is 7.9%. So now I'm actually doing a little bit of a sleight of hand, and I've been deceiving you. And so this score of 0 to 100 is not your risk of having a heart attack or developing cardiovascular disease. This is the ESPN win probability for football games, right? And this is data drawn from Super Bowl, I don't know what it is, 51. It's my last kind of happy, well, I guess there was another Super Bowl. We're spoiled, but this was the famous Falcons Super Bowl. And the point is that 7.9% point estimate hides a lot of activity, hides a lot of drama. And at that point, it was 28 to 20. And I think if you're from New England, you thought, boy, the win probability might be higher than 8%. And there are all these elements, the strip sack and whatever, that go into this trajectory. It's very dynamic. And so what if instead of watching the game, this is how you watch the game? And so you got, baseline stats about the two teams, and then in-game statistics about how much time was left, what down is it, where is the football? This is a crude analogy, but I think there's some similarities to how, the measurements that you clicked on in the poll give us, pretty good estimates of risk, but miss a lot of the action. And so these are episodic. They require a clinic or hospital visit. They incompletely capture biologic complexity and the dynamism that goes into disease development. And we exist in this world where silos wall off clinical care from research. All of these barriers, therefore, limit access to clinical care, limit our ability of different kinds of individuals to monitor and be empowered in their care, and limit ability of all sorts of populations to participate in clinical research. So then our last poll for the moment, could you launch the next poll, please? So have you ever participated in any kind of clinical research study, or have you contributed some of your data to a clinical research study? We'll let that run. Okay. All right, so we can, let's close the poll now. So this is actually better than I thought. So 37% of you have contributed to a research study. That's actually quite good. We know, even from, conditions that are, let's say an individual cancer type is much rarer than, say, cardiovascular disease. The estimate is only about 5% of those patients will participate in a trial. This group, I think, is a relatively active group, and I applaud you for those efforts. So we can close that poll. Thank you very much. And I am going to draw, this goes from, this is a slide from my wife's world as an oncologist, and I want to draw the analogy to cancer. This is a diagram of a patient's tumor, and the different colors reflect the different proportion of her tumor cells that carry different mutations, and the mutations are at the lower left. And so you can see at different points, the yellow grows, and then with treatment, the yellow and the pink go away, and then the blue gets big, shrinks a little bit, and then the dark blue takes off at the end. How do they know this? They know this because as they treat, which I've indicated in orange at the top, they're taking biopsies, right? The biopsy timings are in blue at the bottom, but the biopsy is a substrate for research. It can be cultured in cells, it can be analyzed for genomic changes. The biopsy is a physical embodiment of a culture and workflow in cancer that does not exist in heart disease. It's the physical embodiment of a connection between patient care and research. And at One Brave Idea, that's what we're trying to encourage. If you're doing sequencing is sequencing. The phenotype, the measurement is just DNA sequence, and it doesn't matter what disease or what kind of patient, it's just DNA sequence. Right now, phenotypes or measurements are kind of boutique for different diseases. We're interested in making a more disease agnostic approach to phenotyping so that it is easier to apply a set of informative phenotypes across different conditions, across different diseases, in academia, in hospitals, in drug companies. And we've also are taking kind of a Google Maps approach. From Google Maps, you can look at geolocation information at different levels of resolution. So you can see a photo of the bagel store on the corner and pedestrians walking by, and you can zoom out to, city streets and even see traffic and even zoom out further. And so in One Brave Idea, we're undertaking phenotyping by analogy at different biologic scales. Certainly genomic measurements, but molecular measurements, whether it's RNA sequencing to look at transcription, the gut microbiome, cells, like actually taking people's cells and using that as a unit of measurement, and a host of behavioral and physiologic measurements, which we can do using mobile digital technology. And the goal is to get more informative phenotypes that are still at an acceptable level of burden, and to leverage phenotypes which might be collected anyway when people are going to the doctor, so that you can start to unify patient care and research. The other thing I just want to point out is this vertical axis, which I've said in the diagram of perturbations. And so in many cases, what we'll do is, let's say we'll take cells out of a patient, we'll do some assay, and then we'll perturb those cells. We'll treat those cells with one of 30 different things, and we repeat the assay. And there's a lot of reason to think that this perturbation, right, it's like a kick to the system, and you can learn a lot from seeing how the system responds to that perturbation. And it's a way of getting more informative information out of the same assay. An analogy might be, if you think about the Hollywood squares of all the people on this Zoom call, and someone asked me, gee, who are these people on the Zoom call? What kinds of organizations are they from? What's their background? Who's the audience? And I could look at you and I could say, well, how formally are you dressed? How old are you? Are you wearing glasses? Whatever. And I could take a whole bunch of measurements, or I could ask everybody on the Zoom call 10 questions, right? Same 10 questions for everybody. Obviously, that's going to be more informative. And that's an analogy for the approach we're taking to get more out of these phenotypes. To illustrate how we can get, use better, make better use of data and tests from the hospital, here are a couple of examples. And so that machine on the left, that little white box, that's the same machine that generates your complete blood count, the CBC, right? Every hospital, many clinics in the country, in the world, has a machine like that or something very close. It turns out that machine generates a ton of extra information about your blood cells that it discards, keeps in its memory and never uses as it's reporting out the CBC. And Rahul Deo and others are leading an effort to extract the information from that. So it doesn't require any other work, right? If someone has already said, we need the CBC, and it goes into this machine, the normal result is spit out for patient care, but there's a ton of additional cellular information, again, with and without perturbation. The other thing on the right is that we can also take cells from people's peripheral blood and do assays on those. Here's another example, is echocardiograms, right? So again, for various reasons, people will come to the hospital and they'll have an echocardiogram. Largely, those echocardiograms never leave the databases of that hospital system, and it's used, and someone will track the ejection fraction or the mitral valve, mitral regurgitation or something like that. One Brave Idea has developed, AI automated phenotyping of echocardiograms, so that you get not just what we read out and what we look for as cardiologists or doctors, but all sorts of other phenotypes, some of which we don't fully understand, but they are different in interesting ways, and they can help us identify new patient subsets. And this is a little speculative, but some of you may have heard of multiple companies coming up with, not a huge box that you have to roll around to do an ultrasound, but a wand that plugs into a mobile phone or a tablet, right? And some of them are even guided by artificial intelligence to help a less trained user get a good image. So now think about a world where it's not just, a few hospitalized patients or outpatients requesting an echo, where echoes can be done much more frequently in primary care offices, maybe in some of these, newer entrants into healthcare delivery, like a Walmart Health or a CVS or a Walgreens, and maybe even at home, right? If those studies go to the cloud with privacy protections and get analyzed by AI, now you're really cooking, right? Now you're talking about phenotyping someone's heart in a totally distributed way. And so this is, some of these approaches that we've taken, the red blood cell cytometry, assays on people's peripheral blood, automated interpretation of echocardiograms, and then we combine these phenotypes with and without perturbation with traditional genetic approaches, and then try to validate our hypotheses. And we've taken a number of promising genetic leads through this pathway. And so I think there's a pipeline here, right? And we can start to envision a repeatable process, the phenotypes that we've talked about, but we're trying to get a lot of information from, phenotypes that are not space age in their execution and can be done in a mundane routine context. And you can take those through genetics in a way that we think will help couple patient care to research and even therapeutic discovery. And I'm going to close with just, what we hope can be some of the cultural things and some of the cultural implications we're trying to see if we can't accelerate ourselves. If you go to Google Images and you type in clinical research participant, I've selected a few of them, and this is what they look at. They're all very earnest, but they're all in clinical settings. They're all in hospital settings, right? And you don't see any photos like this, but why not? I really think for cardiovascular disease to reach kind of the next level of innovation, we need to have marathon runners, people who just are leading their lives, eating what they eat, trying to exercise as they can, et cetera, have a seamless, unobtrusive, non-burdensome way to participate in clinical research as they're living and as they're working outside of the hospital walls. And I think as we think about that phenotype stack and incorporating digital, for time reasons, I didn't go into digital assays, but I think with, routine blood draws, using cloud and AI and some digital tools, we can start to make progress towards that. So I'll close with these are the kind of questions I began with. These are the questions that we're dealing with and thinking about hard at One Brave Idea. And hopefully I've started to paint a picture of how some of our work is advancing science, but taking on some of these interesting cultural questions. And I think I'll stop my remarks there. Thank you very much for everything you do on behalf of the AHA, and I'd be glad to take any questions. Oh, wow, there's 6. Jay, you're muted. Good.
You'd think after a year of doing this, right, I'd learn where the unmute button is. But the first question came in, is it worth having the polygenic test score? Shouldn't we all be exercising and eating healthy anyway?
Yeah, so there's a lot there. I mean, yes, we should all be doing the right things, and that goes to, the value does depend on, what is the juice that comes out of the squeeze, right? I guess what my hope is that as our understanding increases as to why what are some of the mechanisms underlying these high polygenic risk scores? Maybe it's inflammation. For some people, it might be this new phenomenon of CHIP, clonal hematopoiesis. But if we can have a therapy or even a biomarker that is more closely coupled to the polygenic risk score, or closely coupled to any biomarker, take it away from polygenic risk scores, I think that can help. Some people have said in just casual conversations, the downside of something like a polygenic risk score is that it's immutable. Whereas if I work hard and I do clean living, I may see my blood pressure improve. I may see my weight improve. And that's a powerful part of the feedback loop. So I think in an ideal world, a polygenic risk score might be coupled with, as science permits, some other kinds of biomarkers that can help people track. There's a broader question of, in general, genetic risk and how useful it is. How do people think about it? How to incorporate it into clinical practice? How should it be paid for? And that last issue does a lot of lifting in terms of how much things get adopted for. But I think these are great open questions.
Second question, the phenotypes tell us a patient subset, but can it really tell us how likely we are to be at risk for a cardiac event?
Yeah, so I think the the idea is that first we identify components of that phenotype stack that are informative, and then it's the relatively long, arduous process to confirm our scientific findings in broader, diverse patient populations and see the effect on outcomes, right? So whatever path that blood pressure needed to be associated with risk of stroke, that's what we would have to go through with these phenotypes. And that's why nowadays there are some pretty powerful data sets where you have electronic health records of people coupled to, for instance, some consented blood samples with DNA genetic information. And so there are some tricks we can do to make that journey faster. But ultimately, we need to go from a patient and what we know about their patient's outcomes to these novel phenotype stacks. It's a great question.
So how much will the work being done at OBI be able to help shift more towards a wellness model, perhaps, instead of a sickness model of health? Does that make sense?
Yeah, absolutely. When Callum put, at the edge of wellness in his title when he applied LOSO a few years ago, that was in his mind. The idea would be if we can find biology that supports detection earlier, before things like blood pressure or lipids or blood sugars go awry in middle age or so, then there's a better opportunity for wellness. Again, in theory, a polygenic risk score exists at birth. You could start to focus on wellness preventive measures in theory if you had a very alarming polygenic risk score, subject to all the caveats we talked about. But I think, to build upon the edifice of risk factors of middle age is how we can emphasize wellness. And everybody has their own systems. There is this thought that if you use, maybe it's a digital solution that can help a family, that can help, we're doing a pilot project with school-aged children. And so there may be tools that can engage people earlier, both, educating about wellness, but also contributing data. So these are some of the things we're thinking about. But but absolutely, that is potentially one of the benefits of this kind of work.
You mentioned getting more people involved in research. How might we go about getting more people involved along your type of research to participate?
Oh, to participate in some of these studies? Yeah, so that's a very interesting question. So right now, many of the studies are reaching out to people in this hospital area. As an example, one thing we're thinking about is you want to be able to engage with people outside of the traditional way of being based at, an academic hospital and then trying to spread the word or using that existing network. And so some of these plans are still in development. But I mentioned, for instance, we're looking at other ways of engaging people. So we did a pilot with a school in the Boston area, and thinking about, can you come up with a kind of curriculum and mini research study in a box that is generalizable, that can go to schools of different socioeconomic groups across the country. We're thinking about, and we'd certainly welcome partnerships, the employer-employee relationship. That's a long-term, often engagement, again, with appropriate safeguards. That is a way to also scale research. Obviously, it's a little tricky, especially now. I think the companies are thinking about a lot of other things. But if people have ideas about, ways that employers and employees could each derive value from an engagement in a research project with One Brave Idea, we'd certainly welcome that. And then the last thing I'll mention is kind of consumer platforms. Some of you may have heard of the Apple Watch being involved in a study. Dr. McRae is leading another study that uses the Apple Watch, right? And in that case, this is a far cry from when you post a sheet of paper with like little tabs you pull off to participate in a research study. You recruit for a research study through the App Store. And that's like a whole nother powerful way to recruit and engage. And as I alluded to, you have not just Apple and Google Android, but you have, companies like CVS and Walgreens and Walmart and Amazon with potentially very powerful networks. And I think we'd love to explore any of those kinds of novel engagement partnerships.
Next question, is the polygenic risk score more valuable for any particular ethnicity or for those that have a family history of heart disease?
So that's an important question that there's a lot to talk about. I think it probably has been looked at. I'm not aware of specific data on polygenic risk scores in specific ethnicities. In genetic research in general, there's more work to be done. Certainly, most of the headlines you have heard about in genetics are based on studies where most of the participants come from a European or related background. There are some efforts in the United States and across the world to be more inclusive. And that speaks to some of those access barriers to care and to research that I alluded to earlier. And it's an ongoing problem, but fortunately, I think it has risen to the surface and people are actively engaged in that. And there are a few examples, for instance, in diabetes, where there is a mechanism that predisposes to risk of diabetes that's really only common in Latin America. And if you did a study in the US or in Europe, you wouldn't see that genetic signal. But if you look in the right population, you can find it. And it's a totally novel mechanism. So it's important.
We're coming to the end of our time. And so I would ask you, I started with the Heart Association 20 years ago, and and learned of the unique history that Boston plays in that story with Dr. White, living in Roxbury, really called the father of the Heart Association, some of the work you were doing. And we're coming up on our 100th anniversary in about 4 years. And so it's taken that long to get to hear how might what you're doing there at OBI really transform this next century, if you will, of heart care and heart wellness before its care.
So, that's a fascinating question. Paul Dudley White, I feel like it was kind of a one-man embodiment of engaging with stakeholders across the ecosystem, right? I had that slide earlier in my presentation. He was a practicing clinician. He was a researcher. He was a public health advocate urging lifestyle modifications. There's a bike path along the Charles River named after Paul Dudley White. His work creating or founding the early days of the American Heart Association created this organization that has very broad, audacious goals, right? And so we wouldn't compare ourselves to Paul Dudley White, but I think this notion that, with the finances being what it is in our system, with issues of access and equity having renewed prominence and a deserved prominence, it's incumbent upon scientists, if they really want to speed adoption and impact of their work, to think with intentionality and early on about the stakeholders and the ecosystem. Our One Brave Idea project is relatively unique, I would say, in that, these considerations were part of the mandate, right? I think in a traditional research grant mechanism, you focus on the science, and there's a paragraph of public health significance, and no one's really called on that. But this is something that we take very seriously, and hand in hand with the scientific work on genetics and phenotyping and artificial intelligence, we're thinking hard and engaging with stakeholders about that. And it's no accident that, I don't know if my screen is mirrored, but it's the AHA who had the vision to take this audacious project on. But we have, unique, powerful sponsors with a tremendous amount of expertise and reach in Verily, AstraZeneca, and Quest Diagnostics. And that also gives us ideas and gives us capabilities that are quite unique. And so maybe that's the answer, right? That Paul Dudley White thought about the ecosystem and acted across many stakeholders in the ecosystem with great humility. It would be wonderful if we could kind of do some 21st century version of that.
Well, I would say from my limited knowledge, you're on your way again, but I love the whole ecosystem approach that you talk about, all that goes in. But thank you so much for your time today. And for those of you who joined and are participating, thank you for your time and your support again, and the many, many ways that you engage with us to ensure, as Dr. Shaw was saying, that everyone in Boston has an equitable chance for a healthy life, for their longest life possible. We appreciate your support and hope you have the rest of a wonderful Thursday. Take care.
Thank you.